It's my great pleasure to introduce Xiaojun Tian. Xiaojun did his PhD in systems biology from Nanjing University and his postdoc at Virginia Tech and the University of Pittsburgh. Since 2017, he has been leading his research lab at the School of Biological and Health Systems Engineering at Arizona State University. His lab's research interests are in employing quantitative experiments and mathematical modeling to understand fundamental problems in synthetic biology. And in keeping with that theme, the title of his talk today is Design, Predict, and Build to Understand and Control Circuit-Host Interactions. And with that, I pass the control over to you, Xiaojun.
Yeah, thank you very much for the kind of introduction and of course the invitation. I feel very excited to have a chance to talk here to share some of my stories in the field of the synthetic biology. So this is my lab. So our lab will do quantitative biology. So we are very interested in system biology and synthetic biology, nonlinear dynamics, gene circuit, and renal disease genetics. So we usually combine mathematical modeling and quantitative experiment to try to understand the basic biological science or the complex disease like the cancer, renal disease. So I studied the synthetic biology research actually in the second year of at ASU. Today I will talk about some stories about these projects. So since I was a PhD student like 15 years ago, I have been following the achievement in the field of synthetic biology. And even I didn't do any related synthetic biology project before I started my lab. There are many remarkable achievements, including but not limiting to some examples listed here across the gene circuit, by computing and by the diagnosis, the cancer therapy, the living materials, drug synthesis. Right? So, and here is engineering principles for the synthetic biology. So we all know this, that we have to go through a design, a build, and test cycle for each synthetic biology circuit. But I didn't know how many cycles we needed to complete one project in my lab until I saw this figure. This is a simplified version for the process of a project to produce a methanic acid. Right? As you can see, from the initial idea to the final system that works, goes multiple rounds of design, build, test, and design, redesign, build, and test cycles, right? So that is one of the challenging for the synthetic biology project. So that means we needed to go through many cycles of this design, build, test until successful, right? And another challenging is we have really high ratio of very low ratio of success. So several in the earlier this year there is a Twitter. I'm not sure if you saw this Twitter or if you voted or not, but majority of the vote says if you want published one synthetic biology project, usually more than 2 is failed, right? So actually, the poll was not designed very well because many wanted to vote 1 out of 10 to 50 instead of just 2 or plus, right? So it's really a challenge to make one circuit system work, right? So the success rate is very, very low. Another challenging is even maybe the circuit is working very well in the well-designed controlled lab setting, but it is very challenging to make it translatable in the outside lab scenarios. So the problem is if we move that to outside lab scenarios, many things are changed. The growth conditions, the temperature, or other factors are changing, right? So the circuit may be failed. So here is a design principles. So modularity, right? So for the modularity, we expect to say if we characterize and test each small module and make sure this small module will work, and we expect to say the whole complex circuit will work if we plug in this small module together, they will work as well. So the input output behavior will be the same, right? But actually, most of the Tian circuit don't function as expected when the parts or the module are assembled, as mentioned by Rose as well in the morning, right? So what is the problem? So there are many factors, problems. So maybe because of mutation, because of many other things. One of the problem is we've interested is about the interaction between the cis engine circuit and the host cells, right? So when we put this whole cis engine circuit into the host cell, the E. coli, yeast or the mammalian cells, it causes burden because it will use many resources that are generated for the cell itself, like the iron polymerase, the ribosome for translation and transcription, right? And that causes burden to the cell. The cell will grow slower, and you will know if the cell grows too fast, that will also cause dilution to the cis engine circuit. Right? So we have a growth feedback, and if we have multiple modules in the cis engine circuit, the multiple modules will compete for the limited resources and also will affect each other inside of the circuit. So we know this because we got the similar problem in the project. So I want to introduce the project in my lab. So our project started from work I did during my postdoc. I worked on a process called epithelial to mesenchymal transition, and I built the first mastermind model for this process. It's basically it's a cell phenotype transition from epithelial cells to the mesenchymal cells. So the epithelial cells are cells you can see from this diagram. A cell connected to each other because of different types of adhesions and junctions. They don't move. They form our organs, tissues, right? So they have a high level of Egan-Herring, that as a marker. But the cell can become individually and lose all of the connections and adhesions, become invasive, and that is the mesenchymal cell. So the transition from epithelial to the mesenchymal cell is EMT, and the transition from the mesenchymal to the epithelial, that is MET. So it's a cell phenotype transition. I built the first mathematical model based on this gene-regulated network. The input is exogenous TGF-beta, which will trigger the transcription of snail, and we have snail protein, and the snail will be promoted the next transcription factor Z, and then both snail and ZEB will inhibit Egan-Herring, that is the marker for the epithelial, and promote the Egan-Herring for the mesenchymal. And you can see here we have a double negative feedback between snail 1 and miR-34 because snail 1 will inhibit miR-34. The translation was miR-34. miR-34 is a macroRNA that will inhibit the translation of the snail 1. Similar double negative feedback between the miR-200 and the ZEB, and another feedback from the because of the inhibition of the autocurrent TGF-beta by the miR-200. So based on this network, we built a model, and we, after some analysis, this is bifurcation. And you can see here, actually, there are two Best-Ball switches. We call this cascading Best-Ball switch. And because the first Best-Ball switch here is when we increase the TGF-beta concentration, the cell will be transitioned from the epithelial state to the partial EMT state. And after a second threshold, and then the cell will be transitioned to the mesenchymal state. The first Best-Ball switch is governed by this nil and mir34 positive feedback, and the second switch is governed by the ZEB and mir200 positive feedback. The first switch is reversible, but the second one is irreversible. So that is something I did before, and we verified this mechanism in the experiment as well. So when I started my lab, I wanted to create a synthetic cascading basketball switch, right? So I really want to understand the mechanism of this cascading basketball switch. One of the motivation is that when we build a model for the EMT process, we simplified the system a lot. We only consider the two trapping factors, the two microRNAs, one all-occurring growth factors, right? So we ignored many other factors. So if we really want to prove actually the coupled positive feedback is an underlying design principle for the cascading basketball switch. And that's the reason I want to build a synthetic cascading basketball switch. So we quickly came up with a design. The design is simple. We have two modules. And the module 1 and module 2, and both modules are self-activation. So the module 1 will activate itself, module 2 will also activate itself. They both also promote each other. So we have 4 positive links in the circuit. And then we will have one inducer to the module 1. And then if we increase the concentration of the inducer, we expect to say either the cell will be changed from no color to the green, and then to the yellow with 2 modules activated, right? Or the cell will be changed from no color to red, and then to the yellow. So either we work for is a stepwise activation of the cell feed. Here is a design. So we use ROC self-activation for the first module, and then we use LuxR self-activation for the second module. They are still connected because the first module will promote this LuxI that will produce C6 that will be used for the second module. And also the second module will produce ROC that will be used for the first module as well. So we have another module, TED-R, that will tune the connection between the strengths between the 2 modules. So our initial idea is very simple. We say, okay, if we make the first step, we need to make sure the first module is the basketball switch, right? The ROC module is the basketball switch. And the second step, we make sure the LuxR module is the basketball switch. And if we tune the connection between the 2 modules, okay, we can achieve this stepwise cascading cell feed transitions, right? So actually, it's not that easy. Later, we found that it's very challenging. And here comes the story about that. The first work we published is about the interaction between the cis-gene circuit and the growth, host cell growth. There is a growth feedback between them. So, and this is a story about the first module, the ROC module. And we tried very hard to make sure the first module is basketball switch, right? And we test first we test the promoter is a keypad. We test the promoter strength, and here the results show that the curve is ultra sensitive, right? You can see from here, the promoter is good, is ultra sensitive. If we close the loop to make the circuit is a self-activation, using the model based on the data from this promoter data fitting, we can predict that this circuit should work as a basketball switch, right? Depending on the initial condition, the circuit can be activated at some threshold of the inducer arabinostoles. If the initial condition is on, like the red curve, it can be maintained here for a very large range. So we could see the basketball region here, a very broad basketball region, right? So, and then we put all the circuit into the cell. First, we measured the distribution of the cells of the GFP, right? And when we increase the TGF beta, what you can see from here is the cell change from no GFP to high GFP levels. There are clearly two population in a cell. That is a very good sign for the best stability, right? This bimodal distribution. That's good sign, but that's not enough to prove this is bestable, because maybe the ultrasensitive circuit could also have a bimodal distribution. So we have to measure the hysteresis curve, and we needed to do two experiments. One experiment with the initial off, so we use different concentration of albendazole dose, and we measure the percentage of cell in the on state. And then another experiment is we trigger the cell with a high dose of inducer to trigger cell into the on state, and then dilute the cell into the media with different concentration of the inducers. And but what we can see from here in experiment, that there's no difference between the two curves. So there's no difference. It doesn't matter whether the initial condition is on or off. They're almost similar, right? So it's not consistent with the prediction. The prediction, we see a bestable region here, but there's no bestable at all, right? So, and we think about this problem, and we change the promoter and change the gene as well. We try to what makes the circuit work, but it's all, we got all the same problem. So there's no hysteresis. So, and then we think about our particles. So, and our particles is with that for the red curve. There's nothing wrong with the black curve, initial off curve. For the red curve, we dilute the on cells, the GFP hash cell, into the fresh media with various dose of the arabinose dose. And I think maybe that is the problem. And then we measure the dynamics of both the cell growth, the cell density, as shown here, that is OD and the GFP. Study from high GFP level, and then we dilute the cell into the fresh media. We see a quick decrease of the GFP, and we almost lost the GFP, right? So that leading to the loss of the memory. And then the system reached to different level of different steady state levels depending on the inducer dose. In most of the dose, the cell didn't recover back to the original high GFP state, right? So the reason is that because after we dilute the cell into the fresh media, the cell will grow very quickly. So the cell will dilute the GFP level, the circular gene expression very quickly. That's the reason we have a very quick drop of the GFP here. And also you can see from here, you can see that when the cell divides, the GFP is gone very quickly, right? So, and then we think we needed to consider the host cell growth into our model, right? Our model is simplified. We didn't consider cell growth. That is a big problem. So we consider cell growth into our model. So as you can see here, in addition to the regulation in the circuit, the gene expression, the whole circuit will cause burden to the cell growth that will inhibit the cell growth rate. And the cell growth will also cause dilution to the gene circuit. And then we reverse our model by adding the equation for the cell density, this second equation, and that also consider the cellular burden because of the gene expression. This part will reduce the growth rate, and also this term is for the dilution of the gene circuit, ROC. And after we include this growth feedback into the model, and then we can see that with the initial And for the low concentration of the inducer, the memory is completely lost. Now we can understand why we see that the quick drop of the GFP in the experiment. Right? So to see the mechanism clearly, here we did another analysis with what you can see from here, we have two curves, the red and the green curves. That is the production rate and the degradation rate of the circuit. And we have three intersection points, and this one is stable steady state, and this one is stable steady state. This one is unstable, and this is off state, and this is unstate. So suppose the cell initially is in the unstate, and then we dilute the cell into the fresh media, the cell will grow. And when the cell grows, it will dilute the ROC, the circuit expression, right? And you can see here, we consider the 4 cell divisions. And the cell start from here, the dilution, and the cell try to recover back. And go to second cell division. And you can see here, after 2 cells division, the cell already crossed the separatrix. After cross the separatrix, there's no reason the system will come back. You can see the curve, the arrows actually goes to the left, try to converge to the off state. That is the reason why this self-activation circuit is so sensitive to the cell growth, right? That's the reason we didn't see the hysteresis at all in the experiment. And how to solve this problem? We came up with a simple design. If we stop the cell growth, and maybe we can see the GFP level maintained, right? So we diluted the active cells into the conditioned media, that is from the stationary phase. We expect to see the GFP level maintained well. In this case, we can stop the cell growth and its mediated dilution. And you can see the data from here, the cell didn't grow and the GFP level maintained very well. And finally, we can see a very good hysteresis curve. The cell memory is maintained very well if the initial is on, and we verified our original prediction by removing the growth feedback, decoupling the growth feedback, right? And no, that's not the end of the story. We now think about the first C17 circuit, toggle switch, right? In toggle switch, we have 2 genes that will inhibit each other. That is also a bicycle switch. And in 2000, it is published, and in their, we checked their protocol, and we found that, okay, they actually dilute the cell every 6 to 8 hours in order to maintain the cells in the log growth phase. And the growth rate is actually constant. And we want to test the toggle switching with our circuit, right? So, and of course, there is a growth feedback between the toggle switch and the growth rate as well. So we also dilute, we trigger the cell into the on state. We have high GFP, and then we dilute the cell into the fresh media, and then we measure the GFP levels. And you can see here the GFP level decreases very quickly. It's very similar to the self-activation circuit. But for all the dose of the inducers, even without any inducers, we can see the system all recover back to the original high state, right? Even we have a very large drop of the GFP, but the system somehow knows where they are, and they can recover back. And of course, if we dilute cells into the conditioned media, the cell will not grow. The GFP level maintained very well. And we want to see, okay, this toggle switch is so robust to the growth feedback, and it doesn't matter if we dilute the cell into the fresh media or into the conditioned media, right? So we all see the hysteresis, right? So what is the mechanism? Why this toggle switch is so robust, right? And we did a mathematical analysis, right? As you can see here, the left panel shows the knuckle analysis. We have 2 variables that is like I and theta R. We have 2 genes that will inhibit each other, right? So And we have two equations, ordinary differential equation. And then if we set each equation equal to 0, we can plot the curve between these two variables. And this black curve is not clean for the lag I, and the red curve is not clean for theta R. And also we have three stable steady states and two stable. And here this is lag I high state, that is on state for us. And then this stable steady state is off state. And then the separatrix is here, is dashed line, and this is unstable steady state. Okay, now let's do the simulation by setting the cell at this on-state, and then give full cell divisions and see how the system goes, right? The initial here, when the cell divide, you will understand because the cell will dilute both, the cell growth will dilute both variables, both theta R and lag I, right? So the arrows will go down to this to the left bottom corner, right? So, and then the system will try to recover back, and then have the second cell division and try to recover back. And after full cell division, you can see that the cells are still in this domain. Even the lag I level is decreased from 40 to 10, right? It's still in this domain. And then the cell reach to the stationary phase, the cell stop growing, the cell, the circuit knows where they are recover back to the original high state, right? So that is a mechanism. So here, for the self-activation circuit, RHC circuit, it has a one positive link. And for the toll switch, it have two negative link, right? The only difference is the topology, they are different, right? But they both of them are bypass switch. So that's the reason this one is very sensitive to the cell growth, but this one is very robust because this separatrix and the dilution direction is perpendicular to this separatrix. But for this one, this dilution direction is more parallel to the separatrix. So that's the reason after several rounds of, it's not easy to cross the separatrix, right? So that is the first project. We get a conclusion that interference of the circuit function by the growth feedback depend on the network topology. So this is we use a simple circuit, but that should apply to many other circuits. As long as you use a host cell, you are not using the cell-free system. We need to really consider the cell growth into our system, and we really need to consider the circuit topology into our design. That is first work. And following that work, we did another following simple project by just changing the nutrition level, because this I did during the COVID, so we have very limited access to the lab. So, and we did this experiment, but when we change the nutrition level, so in the first project, we use 100% IOB, but if we change the nutrition level to 20% IOB, and we see the GFP level decrease similar to the first project, and then goes up to a very high level, and then decrease, relax to the stationary phase, right, steady state. So it looks like there is an emergent depth oscillation here. That is very interesting, right? This is a toggle switch. This is a festival switch circuit, but it shows oscillatory behavior. So we tried to use the same model to fit the data, but it failed. And we mathematically proved that this model is not able to fit this data at all. And then we revised our model by including this ribosome regulation. The production of the regulation will depend on the cell growth, because when the cell grows fast, the cell will produce more and more ribosome for all of the transcription and all of the translation necessary for the cell growth, right? But the ribosome will be used by the circuit and also will be used by the growth. And of course, we have the dilution from the cell growth. right? So we use this model, we can fit the data pretty well. And then we did additional analysis by changing the nutrition level systematically. So in the model, we changed the Mmax, that is the maximum number of the cell density number. Cell will be saturated at this number, and that is determined by the nutrition levels. Okay, and we predict that in some range of Mmax, we could have oscillatory behavior. We have a peak and we have a valley. The valley continuously be lower and lower when we increase the Mmax, but for the peak actually is largest with moderate of levels of Mmax. That means we can see a very large oscillation with a poor nutrition or moderate nutrition level, but not in the high nutrition level. And we verified with experiment by using different fraction of RLB. What you can see here for the 5%, 10%, 20% RLB concentration, we could see a very large peak overshoot for the GFP. And this you can see the density max is also increased with this RLB. Is very consistent with prediction. And these students who also continued this topic by exploring the growth feedback to the circuit, and he first fitted the data that is from a science paper from Terry Hua's work. This data shows how the growth rate depends on the fraction of the heterogeneous gene expression, the C7 gene expression, right? And the paper claims that this kind of linear relationship, but my students fit this data and found that actually there is some nonlinearity here. And we use a Hugh function to fit this. What you can see here, actually the Hugh coefficient is larger than 1. For some nutrition level, it's almost close to 2, right? So that is suggesting us ultra-sensitive growth feedback. Euro we consider as a linear. So the student want to understand if the growth feedback is ultra-sensitive, that can change our steady state number of the C7 circuit. And he applies that to 3 different circuits: the simple circuit with no feedback at all, and a second circuit self-addition, and a toggle switch. And for the simple circuit without any feedback, and we can see if we increase the ultra-sensitivity of the growth feedback for some level range, we could see 3 steady states that suggesting a stability, right? And this is a growth. This flat curve is a production rate because this is constantly produce. The promoter, you can consider that is constituted promoter, right? And but the dilution and the degradation rate depend on the level of the circuit. If the circuit is level X is very low, I mean, then with the increase of the X, it's more like a linear. It's more like a degradation dominated, right? But if the this is both the degradation and the maximum growth rate dominated, right? If the gene expression is very, very high, and you can consider the burden too much and the growth rate will be very low. And here, this curve is more like now we don't have growth rate, cell growth, and the dilution degradation will be much smaller. And in between, we can see the curve is more like this, is a single mode curve, and that is the reason we can see the 3 steady states, 3 intersection points between this curve and the production rate. That's the reason we could say this bestable region, bestability, right? If we extend expand this analysis to the self-activation circuit. Now the production rate is not flat. It will be like this flat curve, right? It will be dependent on the circuit itself, gene expression itself. After some threshold, this gene expression could be changed from very low to the high, and then we have 5 intersection points that suggest this could be tractable, right? And if we apply that to toggle switch, we can see pitchfork bifurcation in terms of this growth rate, right? If the growth rate is large enough, we could see a tractability here as well, right? So that means if the growth feedback is ultra sensitive, maybe we could say multiple steady state, right? That ultra sensitivity of growth feedback could depend on the as you can see from this data, could depend on the nutrition growth conditions. The second project, actually, I want to introduce a major project I want to introduce is about the resource condition. We have 2 modules in circuit, and we found that winner takes all resource condition. This is quite interesting. And the idea is when we have this circuit, we got the problem, and I read this paper, and no, this is a book from a doctor, 7 years old, and no, it's about the competition of the chair between the 2 friends. One side, this is mine, and the other side, I started that I'm sitting in the chair right now, like that. So, and also I read a lot of papers published in the field about the resource condition, right? But our result competition behavior is kind of different. I will show you quickly. So here is the design. I already told you about this cascading bicycle switch circuit design, right? We have 2 modules. For each module, we have a self-activation, and both modules will promote each other. We have just 1 inducer, right? And the theoretical analysis show that this should be a good design for these cascading bicycle switches. And you can see here, we can consider the system. We have 2 variables, module 1 and module 2, and we can do Knuckling analysis, right? We have 2 ordinary differential equations. And the first, we can consider the first knuckling for the module 1 is we take module 2 as a kind of a parameter, and how to see how the module 2 will affect the module 1, right? So if you consider like this way, we will see a curve like this. So when we increase the module 1 at some threshold, we could activate the module 1, right? If we increase the module 2 at this threshold, this is and then we can activate the module 1. Similarly, we could have a knuckling for the module 2. If we increase the module 1 above this threshold, it could also activate the module 2. And because right now in this scenario, the link from module 2 is module 2 to the module 1 is stronger than the link from module 1 to the module 2. That's the reason this threshold is relatively smaller than this one. That's the reason we have 3 steady state like this, and we can see we could expect to see the selfie transition from the no color to the module 1 activated, and then to the co-activation of 2 modules, right? And alternatively, if the link from module 1 to the module 2 is stronger, and we could say this threshold now is shift to the left is much smaller than this threshold, right? Larger than this threshold, and we could have a intersection steady state here, and we could see the selfie transition from no color to the module 2 activated, right? And yellow, right? And then we could see the co-activation, right? So, but what we can see from the experiment is that we didn't, when we increase the inducer dose, the cell changed from no color to the module 2 activated, and we didn't see many cells in the co-activation region, but more cells in the module 1 activated instead, right? So this is so different from the expectation, right? We so what is the problem, right? The self-feed transition path is kind of changed because something, right? And then we read the paper in the literature, and we realize that that is because of resource competition that we didn't consider into our circular design, and we reserve a model by including the resource competition. Now the nuclear analysis is changed because this is the same. This circle is the same when we increase the module 2, which can also activate module 1. But when we increase module 2 too much, we can see a decrease of module 1 and then a switch off of module 1, right? And then we can see 3 different statistics, right? This is consistent with experiment data. And to further confirm this one-tick-sort behavior, we remove the link between 2 modules. Now we have 2 separate basketball switches. The only link between them is a resource condition. And now we fix the dose for the first module. We change the inducer dose C6 for the second dose. And we can see here the cell change from no color to module 1 activated. But if we increase the C6, the cell change from module 1 activation for co-activation. And if the C6 is too large, we could see the winner-takes-all, right? So this winner-takes-all behavior is verified. But here we see the co-activation because now that we have two inducer doses, we added the 2 inducer doses at the same time point, 10.0, right? And the 2 switch can be activated at a similar speed. That's the reason we could have a chance to have both modules activated, right? And then how to solve this problem? We use a division of labor strategy. We use 2 strings, and we put the 1 string in one cell, or module 1 in one string, and the module in another different string. And then you can see here, when we increase the inducer dose, the module 2 first activated, and then module 1 is activated. Now the 2 cells and the 2 modules are in different cells, so we don't see the cell dots here. But what you can see is this module 2 activation didn't disappear, right? So we have a stable co-activation of these 2 circuits. We can never solve the problem, right? So now, and following that work about this resource condition, we are wondering if there is any other control strategy, right? And because maybe for some cases, we have to put all the modules together in one cell. So what is a control strategy we could have? And we read the paper, and many people, many labs are working on that, proposed a different mechanism, right? Controller. So negative feedback controller. So we read the paper, and then we want to see if this negative feedback controller can solve the winner-takes-all problem. And we did the simulation. So here, what you can see, we have 2 separate self-activation, 2 separate basketball switch, right? If under the ideal case, if we don't have resource competition, in the space of the 2 inducer dose, we could see a very large range for the co-activation region because they are independent, right? And if we consider resource competition, and now the co-activation region is shrinked a lot, we have a very small region for the co-activation region. And the reason is that we have a lot of region for the winner-takes-all, because the module will be activated, and then we'll inhibit the other, and this is not good, right? And in the field, we could apply a local controller or a global controller. So for the global controller, so all the modules, the circuit, all the modules will produce something, a control node that will inhibit all of the modules, right? For the local controller, each module will generate one control node that will inhibit itself only. And then we did a simulation, we can see the global controller increase the co-activation region a little bit, but not a lot. The local controller did a better job, right? The co-activation region now is enhanced a lot. But here we propose a different mechanism. So we call this negatively competitive regulatory network, NCR, right? So that is similar to the local controller, but we introduce another competition here. So suppose each module will generate a small get array. These two small get array will compete for the limited decast map for the inhibition. That is good because suppose the module 2 now is the leading, is very close to be the winner, right? And it will generate a lot of small get array, and then we'll use most of the all of the decast map for the inhibition. And in this way, the inhibition to the first module will be released. That will give some advantage to the weaker module, right? And in this way, you can see the co-activation is even larger than the local controller, right? So, and this is also true if we increase controller strength, and the NCR controller is always better than the local controller and the global controller. And we also apply this to the feed forward mechanism, right? So we could have a local controller, and the inducer will also induce some control node to inhibit the module, right? And also we could have a global controller, or the inducer will induce some control node to inhibit all of them, and we could have an NCR by including the decast map, by including condition here. What we can see here, still the NCR is much better when we increase the controller strength. We have the NCR always better than the local controller and the NCR controller. What the difference from the negative feedback controller is that we found a shift of the co-activation threshold from this 1,1 to positive larger than 1,1 for the global controller, but this is negative smaller than 1,1 for the negative for the NCR controller. And if we increase controller strength, we can see the shift is very significant for the NCR and the global, but that opposite for the NCR we have a negative shift, but for the global we have a positive shift. And then we consider that maybe we can use that. And if we consider the consumption, the resource consumption by the circuit, and then we can change this shift from negative to positive, and we could have almost orthogonal system using this NCR feed-forward controller. That is very similar to the ideal case without almost independent, right? Lastly, I don't think I have much time. I want to mention this last word about the resource condition, the role of the resource condition in the gene expression. And we suspect that if we have a resource condition in the system, the translation of one gene or mRNA, the translation of one mRNA will affect the translation of the other. So if we have stochasticity from this process, and they will affect each other. So we analytically solve the noise level for this simple system. We have two simple genes of PGFP. They will compete for the unpolymerized ribosome for the transcription translation. And then, as you can see here, we have three terms for the noise. The first two terms of noise is well consistent with public exchange. The first term is from the birth status of the protein that depends on the mean number of the protein, right? The second noise is from the propagation from the mRNA that depends on mRNA numbers. And then we have additional term that is from the opposing mRNA, right? So that depends on the mean number of the opposing mRNA. So we call this is RC noise. That is so if the GLP could have a noise from the translation of the RP mRNA. So you can see here if we consider that we have a broad distribution that is has a larger region, right? And if we change the resource level, we can see this third term will be increased if we decrease the resource level. This curve will increase. And but the overall the total noise will decrease and then increase. Why is that? Because the resource condition could have a double-edged rule, and if we consider that with unlimited resources, if we can say that actually the unlimited resource is not good, because the unlimited resource case we have a very broad distribution and a very large. The noise is always larger than the case with resource condition, right? Because when we have resource limitation, you can see this curve, the noise for the second term is decreased with what we limited the resource, right? That is the double-edged rule of resource condition. So here is a summary. We demonstrated the topology-dependent mechanism for the growth feedback, and how that will affect our circuit behavior. We demonstrate emergent oscillation that is mediated by the nutrient modulating growth feedback, and we found that the ultrasensitivity growth feedback could drive emergence of quantitative state. And because the resource condition could be one takes all, if we consider nonlinear behavior of the circuit, if the circuit becomes complicated, the resource condition could be one takes all. And we proposed negative convective regulation and there are negative feedback controller and feedforward controller that could be better than a local controller and the global controller. We found a double-edged rule of the resource condition in the gene expression and noise. With that, I want to thank the people in my lab and Dr. Zhang did most of the experiments together with business students from China. Hannah did the modeling analysis for the resource competition. One did the modeling analysis for the growth feedback. Austin participated for the proposed answer controller and participated for the data analysis for the node analysis as well. I want to thank the collaborators, Xiaojun and Dr. Huang online, and also the foundry from ISF and Ansh. Also, will hire me. And that's it. And I'm open to any questions. Thank you very much.
Thank you, Xiaojun. That was a great talk with so many different circuits and so many different things for us to consider in terms of resource competition and growth. And I can see that Ross already has a question. Ross, go ahead.
Yeah, certainly. I found that I don't have enough time, so I have to speed it up. So, but please ask questions. I'm sorry if I talk too fast at the second half of my talk.
That was fantastic. Thanks for going through all that. I found it really interesting. Going back to the, I guess, your introduction in this EMT process.
Yeah.
Now that we've gone through, like, both of us have been looking at like all of these different resource competition, growth coupling, et cetera, effects that can happen. I'm curious what your perspective is on that circuit, and like if you've started to think about what might be like the important couplings in the endogenous EMT network that could be affecting the behavior of that transition.
Oh, you mean if we design some circuit try to control the EMT process?
I'm just thinking about even just in the native context, but if you were to try to...
Oh, okay. I mean, yeah, I think that's a good question. I mean, for the natural system, right, for the EMT process, that's after many years of evolution, I feel like they may have some resource competition problem, but they already involved. You get either try to avoid that, try to take advantage of the resource competition for all of the regulations. And right now, if we add some heterogeneous circuit to the system is the host cell don't expect that, right? So that's the reason we have so many problems because of the resource competition, because of the growth problem. So, and based on that, maybe we can engineer the host cell somehow. If the host cell can generate more extra resources that can be used for our gene circuit, and that can that give us some freedom to design or scale up of the circuit, right? Did I answer your question or?
Thank you for that, Xiaojun. And I was wondering, so with a lot of the designs, the data that you presented, what sort of design rules can we take into account when we want to make, multi-layered circuits? And I'm asking this, well, there is one aspect of course, the resource competition and how it affects growth, but also the fact that this is not constant. So depending on which parts of the circuit are on, there is different load and different resource competition going on. So what, like, for the vision of the future, when we have, perfectly predictable synthetic circuits, what are the different things to think about for those design rules?
Yeah. First of all, we really need to consider this. The host cell growth and the resource competition and all the cellular burden into our design. I mean, I feel like many years ago we tried very hard to optimize the circuit by changing the tuning paths, changing the RBS, the promoter, and to make sure the circuit will work under some specific conditions. But if the conditions change, maybe the circuit will not work very well, right? So now if we understand how the circuit host growth or the cell growth resource condition affect our gene circuit, and we are accumulating more and more design principles, new design principles like the topology, right? So we may want to avoid using the self-activation. Maybe we want to use more inhibition link into the circuit design. So, but that just give us some kind of a hint. So I'm not sure if for a more complicated circuit, is that a good design principle or not, right? So we have to, I think there are a lot of work we need to do to understand this circuit host system, and then we will have more and more better idea how all this hidden interaction will affect our circuit and how to prevent that. So, but anyway, we need to keep this in mind anyway. Otherwise, high chance the circuit will be failed. So, and it will be very frustrated. Yeah, especially for a very new lab who started the project like my lab. So we were very frustrated. We changed this, changed that, it didn't work, and now we think about my career and the career of the students and the postdocs. What should we do? And we said, okay, maybe it's a field for us, it's a field for many labs. It's a general problem, it's a fundamental problem. We needed to understand why, and then we can say about how to solve that. Yeah.
Okay. Thank you. Thanks a lot. Are there any more questions? Is there any questions in Augusta, Matthias? I can see Ross with another question. So unless there's nothing from the audience in Augusta, let's go to Ross again.
Great, thanks. Yeah, I'm sure that we could talk all day, Xiaojun.
Yes.
That I was thinking about, because I'm more of a mammalian person, but it seems like bacteria are so optimized, right, in terms of how they've evolved, that just even minor amounts of gene expression can sort of perturb the cell. And I'm curious how you were starting to think about this in trying to design maybe like minimally perturbing systems or things like that, that would both minimize their impact on the cell and then also be designed in a robust way through like the design schematics that you illustrated today.
Yeah, that is a very great question. And suppose we can't engineer the cell, the host cell is there. We needed to use that, and we have to minimize cellular demand, the resource demand. right? To actually avoid accident. So, but still, maybe we can find some strategy. We can make sure the burden is very minimal, and we can have maybe one module and two modules, but still, when the module try to increase more and more modules is considered into a circuit, at some level, we have to consider this resource condition and the burden. I'm thinking about a crazy idea before. I'm thinking about if we have some level of resources in this cell and some level of resource in this cell. If we fuse these two cells together, we have a larger cell. We could have more resources that could be used for the oxygen circuit. But I feel like maybe that's not an easy project. But if we fuse more and more cells, that can become a more reactor, it's more like a cell-free system. Yeah. So, but I feel like if we can learn more about the natural system, maybe we could get some idea about how to minimize and make sure, and also our circuit will work definitely. Because sometimes when we try to minimize the load, the cellular load to the circuit will not work. We have to use a very strong motor somehow, right, to make sure the circuit will work. And yeah, that's a balance. I don't know. Yeah, but yeah, we can talk about this more maybe later.
Thank you, Xiaojun.